slides if you'd like. Okay. Oops. Okay, so welcome everyone to the McGill Biomedical Technical Seminar Series. So if you don't know, this is a series of monthly talks hosted by experimental medicine, physiology, and pharmacology and therapeutics, and they consist of talks given by grad students for grad students and is meant to show wet lab or data analysis techniques. And so today I'm really happy to introduce Ariana. She's a master's student from Dr. Chen Liang's laboratory at the Lady Davis Institute, and today she'll be discussing various applications of viral vectors. So whenever you're ready, take it away.
Okay, so can everyone see my screen? Okay, sorry, my Zoom is getting a little finicky, but I'm just gonna go with it. Okay, perfect. So hi everyone, thanks for joining me. And thank you, Michelle, for the invite to present today. So my name is Ariana, I'm a master's student, I'm a first year master's student in experimental medicine, and I'm a member of the Liang Lab at the Lady Davis Institute. So my work is focused mainly on virology and more specifically on SARS-CoV-2 and the different mechanisms by which this virus has been evading host immunity throughout the COVID-19 pandemic. And so today I'm really excited to be introducing you to the topic of viral vectors and how to use them for genetic modification. So I can't mention viral vectors and COVID-19 without mentioning the fact that these vectors have actually helped us in our fight against the COVID-19 pandemic. So specifically here, this is an adenovirus vector, and I'll be going into more detail about the different types of vectors that are available later in the presentation. But essentially what this is a virus that lacks the genes responsible for replication, and instead it transports the genetic material for the SARS-CoV-2 spike protein, which is the protein that's on the surface of the virus. So what's going to happen is that when individuals are infected with these vaccines, their cells will receive the genetic information for the spike and then be able to produce that spike protein. And so eventually when they get infected with, or if they get infected with SARS-CoV-2, they'll be protected and it'll have generated an immune response. And so this is just one of the many applications of virus vectors that I'll be going over today. So in today's talk, what I'd like to do is first present you the concept of viral vectors, and then I'll go into some of the different types of vectors that are available and their applications. I'll then tell you how to safely produce these vectors in your own experiments, and then I'll tell you a little bit about how I use them in my own work. So first off, what are viral vectors? So they're essentially modified viruses that are used as tools to deliver genetic material into cells. And this is a very general viral life cycle. It's not for one virus in particular. But what I really want you to appreciate is that every virus has had to develop their own way to infect cells, to get into cells, and then trick these cells to replicate their genome, and then assemble the genetic material and the proteins into new viruses that can go on to infect new cells. So when we're talking about virus vectors, what we're talking about is taking a given virus and engineering its genome to carry another gene of interest. So when these vectors will infect the host cell, what they'll do is trick these cells to produce this new gene instead of the genes that are needed for replication of the virus. So of course, it's easy to see how this idea of using these vectors to essentially infect cells and make them express that we want is a very appealing concept. And although it's not this simple in practice, virus vectors really open up amazing doors for genetic modification. So they were initially developed for use in molecular genetic experiments, so as an alternative to just transfecting DNA into cells. And this is because compared to some of the more traditional transfection mechanisms, like transfection methods like using PEI or lipofectamine, which may not necessarily achieve as high transfection rates in certain cell types, with these viral vectors, you can achieve up to 100% transduction efficiencies depending on the cell types. So this is especially useful in expressing genes in cell types that are more hard to transfect. And of course, these types of experiments where we use these vectors to overexpress genes are still routinely done in many labs. Another thing that can be done is stable expression of a given transgene by using virus vectors that actually integrate their DNA into the host cell genome. So for this, we normally use retroviral or lentiviral vectors, which I'll also be discussing later in the presentation. But this method is a very simple way to produce stable cell lines that express a given gene of interest. And of course, while overexpression is possible with these types of vectors, you can also use them to deliver siRNAs for knockdown experiments. So for in vitro work, compared to using synthetic siRNAs, although this approach presents more biosafety risks and it may also take longer to develop, there's some benefits in that you can use, again, stable integrating virus vectors and stably knock down a given gene. And these vectors have also been used for the delivery of CRISPR-Cas9 genome editing machinery. So like with the other approaches, the virus vector will protect the genetic material, allow it to enter the cells, and then your Cas9 and guide RNA can get expressed. Viral vectors can also be used for more in vivo biomedical research approaches. So for example, to make transgenic animal models. So here, instead of just microinjecting a zygote and then implanting it into a carrier female, you can instead transduce those zygotes. And for these, again, we often use integrating vectors like lenti or retroviruses. And then you can implant them into your female. And this is a very effective way of generating transgenic animal models. So more efficient than a microinjection technique. So outside of research, there's also many roles for this technology as well. So for example, I've already alluded to their importance in vaccine development by mentioning the COVID-19 vaccine. But there's also a lot of work also going on to try and use these vectors in gene therapy and cancer therapy. And I'll be discussing this quite a bit later. But this is just a summary of the three sort of main types of virus vectors that are commonly used in these studies. So there's adenovirus, lentivirus, and adeno-associated virus, or AAV. And here you have the number of ongoing clinical trials as of 2021. So it's quite a large number. And although the different types of viral vectors are different, so they have different properties and they're different from one another, they have to share a number of key properties. So the first one is safety. So often these viruses are modified to really reduce the risk of harm. So often we delete parts of the virus that are critical for replication. And in doing so, we ensure that it can infect the cells, deliver whatever gene we want to be delivered or whatever genetic material that we want to be delivered, but that it won't replicate. And one really big ongoing safety concern with viruses, particularly ones that integrate, is insertional mutagenesis. So this is where an integrated gene can either activate a proto-oncogene or can disrupt a tumor suppressor gene. And this would lead to malignant transformation of cells. And this is especially important when you're using these for in vivo approaches. These vectors should also have low toxicity, so they often have minimal effects on cell viability and cell physiology. And again, this is really important when using them in patients because you want to avoid any adverse immune reactions. They have to be stable, so you want to avoid genetic instability that would lead to random recombination events that would accidentally produce, for example, replication competent viruses. Cell type specificity, so often we engineer these viruses to either have a broad or narrow tropism, depending on what you need your viral vector to do. And we can also pseudotype, where we can alter the envelope protein that's expressed on that virus to allow it to infect different range of cells as it would normally infect. And finally, selection. So these vectors often contain selectable markers that code for example, antibiotic resistance or fluorescence markers like GFP, identify cells that were infected by, for example, treating with antibiotics or looking for fluorescence under a microscope. So now what I'd like to do is give you or introduce you to the different types of viral vectors. For each of them, give you some insights into the way that they're currently being used, especially in clinic. And I'll go into some of the advantages and limitations for the different types so you can really appreciate when you use one versus another. So these are the different types of viral vectors that have been developed to date, with the first four being the most common. But I'll go into each of them in a little bit more detail. So first is adenovirus. This is a relatively large virus. It's around 26 to 40 kilobases in size. And chances are we've all been infected with an adenovirus before because they actually cause around 5% of common colds. And they're very immunogenic viruses. So they have numerous uses in gene therapy, cancer therapy, and vaccines. So again, the COVID-19 vaccine, for example. And so in terms of some of their advantages and disadvantages, because they're quite large, you can insert relatively large genes into them. There's efficient transduction in most cell types and most tissues, and you can get very high titers when producing these viruses. But some disadvantages are their high immunogenicity. And because like I said, we've all likely been infected with an adenovirus, this pre-existing immunity might cause some problems when you're trying to express genes in a patient. And this high immunogenicity actually is possible that it's for the increased risk of blood clots that we are seeing with, for example, the AstraZeneca vaccine, which is an adenovirus-based vaccine. So this is something that has to be considered when using these in the clinic. So adenoviruses, because of this high immunogenicity, actually experience quite a few setbacks in their initial uses in gene therapy. So they were initially used for the delivery of the CFTR gene, which is defective in cystic fibrosis. And what researchers noticed is that with each dose that they were giving patients, the adverse immune reactions became more and more severe. And the use of these vectors really kind of stopped for a while in 1999 with the case of Jesse Gelsinger. So I'm sure many of you have heard of this case already, but he was young, he was only 18 years old, and he suffered from ornithine transcarbamylase deficiency, or OTC. So he had a dysfunctional OTC gene, and it led to ammonia buildup in his blood to potentially toxic levels. And so he took part in this clinical trial that tried to use adenovirus vectors to sort of replace that OTC gene. But unfortunately, after the first dose that he received, it triggered a fatal cytokine storm, so an exaggerated immune response, and he passed away a couple of days later. So this really kind of stopped the use of adenovirus vectors for a while. But despite these setbacks, they've really returned into the spotlight in recent years because researchers have learned how to use this immunogenicity or this kind of exaggerated immune response to their advantage. And so now these vectors are really some of the most commonly studied viral vectors in gene therapy and cancer treatment. So one interesting application of these vectors is oncolytic adenoviruses. So these are replication competent in cancer cells only. So they're modified viruses, they're safe. So essentially what happens is that they can infect normal cells and they can infect cancer cells, but it's only the environment in the cancer cells, so the gene expression profile within these cells, that allows these viruses to undergo their normal replication cycle. And as a result of the normal lytic life cycle of these viruses, they're able to replicate and lyse the cells and kill them as a result. And one success story that I'll be mentioning in the landscape of viral vectors is Oncorin. So this is an AV oncolytic vector that was approved in China, and it's for nasopharyngeal carcinomas. So next up are adeno-associated viruses, or AAV vectors. And as is evident by this image, they're really small. So this is an adenovirus vector here, and then here is an AAV vector. So they're tiny. And one additional note with AAV is that there's different serotypes of these viruses. And they have different affinities for different cell surface receptors. So depending on what serotype you use, it may be able to infect certain cell types and not others. And this has really important implications when you're using them as a vector. But they don't cause any known human disease. So in terms of their use in gene therapy, they're really the leading platform for gene delivery, despite the fact that they're so small. And this is a diagram for around 150 clinical trials that are going on based on the AAV serotype that's used, the primary tissue target, and the phase that the trial is in. And what's interesting to note is that the eye, the liver, the muscle, and the heart, the eye, the liver, the muscle, and the central nervous system are really common targets. But other major organs like the heart, the kidney, and the lung remain largely inaccessible by these types of vectors. But a really interesting approach that's going on to try and get around this problem is to make more clinically desirable capsids. So in order to kind of increase the tropism of these different AAV vectors, researchers have been trying to do capsid shuffling or error-prone PCR to sort of alter what cell types these AAV vectors can infect. So in terms of some of the advantages and disadvantages of these vectors, because they don't cause any known human disease, they're non-inflammatory, non-pathogenic, but like I mentioned, they're very small, so you're kind of limited in terms of what gene you want to insert into these vectors. And because often these viruses require helper viruses to produce, it's hard to get pure stocks of them. But again, another success story in the viral vector landscape. So this is a drug that was approved in 2018 to treat spinal muscular atrophy, and it's an AAV vector. So this is a condition of the central nervous system and peripheral nervous system. So next up are retroviruses. So there's two subcategories of retroviruses, being simple and complex. So simple retroviruses include gamma retrovirus or MLV, which is murine leukemia virus. And then complex retroviruses are lentiviruses. So for example, like HIV-1, the virus that causes AIDS. So when we're talking about a retroviral vector, we're talking about vectors that are derived or that are made from these simple retroviruses. So the genome of these is around, it's a little bit bigger than the AAV vectors, but it's still quite small, so around 7 to 11 kb. And the genome only has 3 open reading frames, so GAG, POL, and EMF. And while these vectors are limited to only infecting dividing cells, they've really found their use in numerous applications in gene therapy, cancer therapy, and tissue engineering studies, so especially studies in bone repair. And on the other hand, lentiviruses, as I mentioned, are a subcategory of the retrovirus family. So these are characterized as complex because their genome contains some additional genes. So it does have GAG, POL, and EMF, but it also has these additional accessory factors. So the most common lentiviral vector is made from HIV-1. And a really interesting aspect of the life cycle of retroviruses and lentiviruses is their ability to reverse transcribe their RNA into cDNA upon infection. And then they can integrate this DNA into the genome of the host cell that they infect. So this is something that we really take advantage of when we're using these as viral vectors, because they really allow for long-term expression of a given gene because it's integrated into the host cell genome and can be expressed. So in terms of the properties of these two virus vectors, what kind of differentiates them is the cell types that they can infect. So retroviruses can only infect dividing cells, and lentiviruses can also infect non-dividing cells, which makes them really useful in studies dealing with, for example, the nervous tissue or cardiac tissue. One really important risk, though, of these two types of vectors is insertional mutagenesis. So I've alluded to this before, where it can activate, for example, a proto-oncogene or disrupt the tumor suppressor gene, but this is something that was seen, for example, in numerous clinical trials. So for example, in one trial, they tried to use retroviruses to treat X-linked severe combined immunodeficiency or XSCID. And what happened was that 5 out of 20 patients that were in this study, so 25%, they developed vector-induced leukemia as a result of this treatment. And I believe 1 or 2 actually passed away. So this is an ongoing risk that still kind of has to be, I guess, dealt with to completely remove the risk. But that being said, these vectors really are still important tools. And one really interesting application of these is for the production of CAR T cells. So this is what lentivirus vectors specifically. So what these are, they're modified T cells that express an artificial T cell receptor for use in immunotherapy. So they're modified to be able to recognize cancer cells and then get activated and destroy cancer cells in a patient. So how they're made is you extract the blood from a patient, and then to insert the CAR gene, you would use a lentiviral vector. So this would allow stable integration of this gene into the T cells. And then from there, expand or select for the T cells that were infected, and then reinfuse them back into your patient. And then those cells would be able to go on and kill cancer cells. So herpes viruses are another type of virus that's often used as a viral vector. And an interesting point about this virus is that it's really big. So it's much larger than the other viruses which I've previously discussed. And these are the that commonly cause like cold sores. Another really, sorry, I'm gonna try to move my Zoom thing here. Okay. Okay, so a really interesting aspect about the HSV life cycle is their ability to establish lifelong persistent infection in a host. So what happens is that when these viruses infect, the progeny virions are going to exit the epithelial cells and then go on to infect neuronal cells. And here the DNA is going to form episomes, so these circular portions of DNA. So it's not that it's integrating into the genome of these cells, but instead it's just remaining latent until some sort of stress reactivates virus formation. And from here, the viruses are going to be able to go on and infect new cells. So this really unique aspect, the fact that the viruses can remain latent and remain inside the host without actually integrating and as well as the fact that the genome is so large, is really something that's inspired researchers to try and use these in therapeutics. So like I mentioned, they're very large. So this is something that's good because you really have a wide you can insert really large therapeutic genes, and they're really good for neuronal cells, so for treatment of neurological disorders. But some disadvantages of these is that they're highly immunogenic, and we still don't really understand how to control that latency on-off. And there's no expression during the latent phase, so this is a really big disadvantage. And in cells that are not neuronal cells, you can only get transient expression of genes. So these vectors have really become promising neurotropic vectors in the last decade. So initially they were used more for local administration, but their sort of design has really improved over the last decade, where now we can actually target them to specific sites or specific cells in the brain. So you don't need this local administration anymore. And so there's numerous clinical trials that have gone on to try and use these to treat disorders like Parkinson's. And there's currently a lot of work now being done to use these to treat glioblastoma, which is an aggressive form of brain cancer. So researchers are trying to use these similar to adenoviruses as oncolytic viruses to selectively destroy tumors. And this is another success story here of a drug that's currently in use. So this is an oncolytic HSV1 based virus that was approved in 2016 to treat inoperable melanomas. So when injected directly into the tumor or the skin lesion, it'll kill those tumor cells. The second to last virus, which I'll be discussing, is poxviruses. And these are the family that includes smallpox and vaccinia, as well as cowpox. And smallpox now has been eradicated thanks to vaccinia virus. So we've used this vaccinia as a vaccine for smallpox. So there's a lot of work that's gone on with vaccinia, and we know a lot about its safety. And they're really big viruses as well, similar to herpes viruses. That being said, these vectors are really underrepresented in the viral vector landscape. So despite the fact that we know they're safe, especially when it comes to vaccinia, and we know that they're able to induce a protective immune response, there's numerous characteristics about their life cycle that doesn't make them the ideal candidate to be used as a virus vector. So for example, their lytic life cycle, because they lyse to cells, it doesn't allow for more stable, or it only allows for very transient expression of genes. So as I mentioned, you can get high levels of expression with these, and you can get relatively large therapeutic genes inserted, but there's some potential cytopathic effects. So the fact that they lyse the cells, and also because these viruses are so large, there's unknown action of a lot of the genes, and this can be problematic when using these in patients because we don't know how they'll react to that treatment. But like adenoviruses, these poxviruses have been explored a lot for their oncolytic properties, and they've also been used a lot in vaccines. So these are the three common types of poxviruses that have been developed and used mostly in trials trying to develop a vaccine against HIV. So the first is MVA and the second is Nivax. So these are two modified vaccinia viruses, but probably the most famous poxvirus vector is LVAC. So this is a canary pox-based vector. And it was the one that was used in the HIV-1 TIE trial. So this TIE trial was the only HIV-1 vaccination trial that has demonstrated any sort of modest efficacy against acquiring this disease. So these vectors they're very promising, but there does need to be some improvements made to their immunogenicity. So researchers are exploring the idea of using different options like prime boosts, co-stimulatory molecules, maybe enhancing the promoter strength of the transgene in order to just make them better suited for, in these. And the last virus vector that I'm going to be discussing is Baculovirus. So this is actually a virus that doesn't infect mammalian cells in nature. It normally infects insects. But since the realization that these can actually infect mammalian cells, they've been widely used, or they're commonly used as a virus vector. So here we can see that they often infect insect larvae and cells, but mammalian cells as well. And they're quite large viruses too. So of course, these viruses don't have very high cytotoxicity because they can't replicate within mammals, but because they normally infect insects, the range of cells that you can infect with these is limited. So these are not the most commonly studied viral vector, but they still do have really important uses in vaccines and cancer gene therapy. So one really important application of these vectors was the development of the Cervarix vaccine. So this vaccine served to protect against HPV type 16 and 18, which are responsible for around 70% of cases of cervical cancer. So this vaccine, what it does is it delivers the capsid antigen of these two human papilloma virus subtypes, so 16 and 18. And it does this using a Baculovirus expression system. So this Cervarix vaccine was actually taken off the market in the US, I think recently. And this is not because it doesn't work, it's just because we have more multivalent vaccines now that prevent against infection of more than just two types of HPV types. So they're more protective, but this vaccine was a very effective vaccine. And these baculoviruses are often used in veterinary medicine to protect animals against different viruses as well. So now what I'd like to do is discuss how to safely package your viral particles for your own downstream application. So in this talk, I'll speak specifically on lentiviral production, because these are the ones that I use mostly in my own work, but the overall workflow is very similar for all the viral vectors. So this is the workflow for producing your viral particles. So it starts with expanding the cell type that you're then going to transfect with the necessary genetic material that's needed to expand your virus. And then this is followed by viral harvesting and then downstream steps that may or may not be of use to you depending on what you're using these for. So for example, this includes purifying and concentrating, titering, quality control. So this is really important for like clinical studies, and then storage as well. So as I mentioned, the first step is cell expansion. So here with most virus vectors, you identify a cell line that's easily transfectable. So often we use 293 T cells because they have a really, really high transfection efficiency. That being said, with some virus vectors, this doesn't work. So for example, with retrovirus vectors, you have to package them in specific packaging cell lines. So these cell lines already express some of the packaging genes and the envelope genes. And so you can't just use regular 293 T cells with these. So often we expand these cells in a 10-centimeter dish so that you have you can transfect large amounts of DNA to increase your virus yields. And you want to see these cells so that they're around 70% confluent on the day of your transfection. And then once your cells are confluent enough, you're ready to begin assembling your virus particles. So when it comes to lentiviral vectors, this involves the transfection of 3 separate plasmids. So the first is a packaging plasmid. And this plasmid encodes transacting elements that are needed for integration of the viral genome, as well as packaging of your viral particles. Then you have an envelope plasmid. So this encodes the viral glycoprotein that's going to be expressed on the surface of your virus, and it's going to be the protein that will mediate entry into cells. And finally, you have the transfer plasmid. So this is the one that actually contains your gene of interest. And often we transfect these in a 3:1:4 ratio, using often just PEI. And then after around 6 hours or overnight incubation, depending on how you do this, you can replenish the transfection media and then just allow your virus to be expanded. And all of this has to be done in a BSL-2 setting. So taking a closer look at these plasmids that are needed for lentiviral production. So I previously mentioned that these virus vectors have to have reduced biosafety risks. So when it comes to lentiviral vectors, there have been 3 generations of these sort of systems that have been developed in order to ensure that you're not accidentally generating replication competent viruses when you're producing these. So when it comes to the first system, this packaging plasmid expresses the HIV-1 gag pol, as well as a number of accessory factors that sort of assist in replication. And you also have this RRE element that sort of regulates the transport of viral RNAs. When it comes to the envelope plasmid, because lentiviral particles tend to be pseudotyped, it doesn't actually encode a lentiviral envelope protein. So instead, it encodes the glycoprotein G from VSV, which is a vesicular stomatitis virus. And the reason we often use this glycoprotein in a lot of viral vectors as well is for 2 reasons. So the first is that it's very stable, so it allows you to get higher viral titers. And the second is that it has a wide tropism, so you can really make sure that your vectors are going to be able to infect a broad range of cells. And then finally, you have the transfer plasmid. So this encodes your transgene, and it also has other elements as well. So for example, LTRs, and it has this size sequence here, which is it's the signal for this DNA to get packaged into your particles. And so here, because the transfer plasmid doesn't actually encode any of these viral proteins, it'll make sure that these genes here are not being incorporated into the virus vector. So as a result, the virus will be replication incompetent when it infects a new cell. So it'll just be there to deliver the DNA or deliver the gene of interest, and then it won't be able to produce more of itself. The second generation was made more safe. So this is because these genes, these additional accessory factors were removed since they weren't necessary for lentiviral vector function. And then the third generation is the most safe. And it's the safest because it contains this deletion in the U3 LTR. So there are certain biosafety risks associated with functional LTRs. So the first is that if you do accidentally generate these replication competent viruses, these LTRs are going to allow them to replicate, allow viral replication. And the second reason why they present a biosafety risk is that because they have enhancer sequences within them, if they integrate next to a proto-oncogene, this is going to be able to activate that downstream proto-oncogene. And this is what was seen with XKID, that case I presented previously with retroviruses. So to avoid these issues, they deleted the U3 region, and they also removed TAT, and they put the Rev on a separate plasmid. That being said, although this is the most safe system, it doesn't give as high viral titers as the second generation system. So for example, in my own work, I use the second generation, but depending on the application, the third generation might also be of use to you. So the next step in the production workflow is harvesting your virus. So this is performed around 48 to 72 hours after transfection. So the first step is you collect the supernatant, which contains the majority of your virus. And then what you want to do is spin it down to remove any cell debris and filter it to ensure that you really remove any debris that's present in that supernatant. And then depending on how much virus you want, you can also add fresh media to your cells and then leave it for an additional 24 hours and perform a second harvest. And if you do choose to do this, it's recommended that you keep your first harvest in the fridge and then add your second harvest directly to it. And instead of freezing it and then thawing it, since freeze-thaw cycles, it reduces your viral titer. And then once you do this, you can either directly use your supernatants for your in vitro infections, or you can continue on in the workflow to concentrate, titer them, etc. So there's numerous ways for you to concentrate your virus, but ultracentrifugation is a really common technique. The way we do this in our own lab is we use a mixture of 15% sucrose, which we add to which we add our supernatants on top. And then we spin them down at very high speeds for an hour at 4 degrees. And this will lead your virus to be in a little pellet at the bottom of your tube. And then you can resuspend that in media or PBS, and it'll be more concentrated compared to just being in a larger supernatant volume. And so an additional step that you can do is titering your virus in order to determine what your viral concentration is. And there's numerous ways that you can do this, but ideally you want to use a method that'll give you a functional titer rather than a physical titer. So the difference is that physical titer will just tell you how many viruses are in your mix, I guess. But with a functional titer, it'll tell you how many of these are actually infectious. So you know how many are actually able to infect cells. So some methods of titering include qPCR and ELISA. You can also use flow cytometry. This is especially useful if your virus has some sort of fluorescence tag. So for example, if you know that your virus expresses GFP, you can look for how many cells are GFP positive through flow cytometry. And what this does is it allows you to detect the multiplicity of infection at which you want to infect your cells. And this refers to the ratio of virus particles to host cells. So for example, let's say your MY is 1, you know that on average, every cell is going to receive 1 virus. And this is the formula that you can use to calculate your MOI for your own experiments. Quality control might also be something that's of use to you or that you may or may not choose to do, but this is especially important if you plan to use your vectors for clinical research. So there's many ways in which you can purify or control the quality of your virus. So this is a table that suggests the step you should take for vectors, but it's similar with lentiviral vectors and other vectors as well. So indicated is the test to be performed, the method you can use to do it, and then some specifications about like the standards that you should be looking for. So for example, in terms of safety, you want to make sure that you don't have any contaminants in your virus like mycoplasma or endotoxins. For purity, you really want to make sure that you don't have any residual cell debris. So for example, you don't want any packaging cells in your virus left over. And you also want to ensure the potency. So before giving your viruses to patients, you want to, for example, make sure that your virus is actually expressing the gene that you want it to be expressing. So you want to do some in vitro work before, just to check that. And in terms of storage, these viruses are stable at 4 degrees for about a week, but for long-term storage, they should be stored in -80. And aliquoted volumes are really preferred for long-term storage to reduce the loss of viral titer through freeze-thaw cycles. So lastly, I'd like to just discuss how I use these viral vectors in my own research. So I'll be giving you a little bit of information about like what my project is and then how I plan to benefit from this technology. So in our lab, we mostly perform our work in tissue models. So most of us benefit from more of the in vitro applications of these virus vectors. So mostly this includes overexpression, as well as for the generation of stable cell lines. So I mentioned that I focus on SARS-CoV-2. So this is the genome structure of this virus. And more specifically, I focus on this protein here, ORF8. So it's a very small protein, but it's very important. So it's part of these accessory proteins here. And it's quite interesting. It has a couple of interesting properties. So for example, it's secreted into the blood. So COVID-19 patients have this protein present in their blood. It's a very immunogenic protein, and we don't know why infected cells secrete this protein. Yeah. Additionally, patients that are infected with a SARS-CoV-2 strain that's deficient in ORF8 have less severe disease, so it contributes to pathogenesis. And it also is able to antagonize antigen presentation through its downregulation of the MHC1 surface receptor. So one thing that I additionally found was that it downregulates another surface receptor, Fc gamma receptor 1a, and overall engagement of antibodies with different Fc receptors on immune cells is really important for immune protection. So while antibodies are able to mediate neutralization of different viruses through their FAB portion, the Fc portion engages with Fc receptors on immune cells, and this will trigger downstream effector responses within these cells. And so for those of you that work in oncology, I'm sure you're familiar with the concept of Fc engineering, where you can engineer the Fc portion of your anti-cancer monoclonal antibodies in order to kind of increase their efficacy at modulating functions like ADCC or ADCP. And this would kind of enhance their anti-cancer properties. So more specifically, the Fc gamma receptor 1a is present on the surface of myeloid immune cells, so specifically monocytes and macrophages. And when this receptor will associate with antibodies, it'll trigger effector cell responses in phagocytes. So this will lead to ADCP, so antibody-dependent cellular phagocytosis, as well as the release of pro-inflammatory cytokines that work to clear the virus. And so this is the data that in which I showed that Fc gamma receptor 1a is downregulated by ORF8. So what I did was I transfected 293 T cells with the same amount of Fc gamma receptor 1a and then varying amounts of ORF8. And what we can see here is that at the highest dose of ORF8, we have around an 86% reduction of 1a. And we have this really nice dose-dependent decrease of this receptor. So this is interesting, but the thing with this is it's a transfection experiment in 293 T cells. So these cells are kidney cells, they're not immune cells, they're not monocytes, and also they don't endogenously express this receptor. So this would not be an ideal model for studying this property further. So the question is, what about in monocytes and macrophages? So of course, a better sort of model for these types of experiments would be THP1 cells, which are a monocytic cell line that endogenously express the Fc gamma receptor 1a. But of course, there's a downside to using this cell line. They're very hard to transfect. So I wouldn't be able to just transfect ORF8 encoding DNA into these cells like I was previously doing, and then study the effect of Fc gamma receptor 1a downregulation. So for these cells, the transduction of ORF8 is instead a much better option. And while I was just transfecting this ORF8 DNA into cells previously, this protein that we have in the lab is encoded in a lentiviral transfer plasmid. So what you can see here is that there's the LTRs, there's the RRE element, and then they have this Psi packaging sequence that would allow this plasmid to get packaged into a lentiviral particle. So ideally, what I would like to do is to produce ORF8 lentiviral particles, transduce them into THP1 cells, and then I would be able to select my cells with puromycin in this case, which is the selection marker for this plasmid. And then what I could do is use these in my downstream experiments, looking at the effect of this protein on monocytes and how Fc gamma receptor 1a downregulation might affect their function. But of course, there's a catch. And this catch really highlights another additional limitation of using virus vectors that I didn't really go into too much detail in throughout this presentation. But this catch is the existence of viral restriction factors. So these are cellular enzymes that exist to block viral replication, and they're really one of the first line of defenses that we have against virus infection. So specifically when it comes to monocytes like THP1 cells, they express an enzyme called SAMHD1, and this restricts HIV-1 and lentivirus infection. So it's responsible for, what it does is it blocks infection early on and prevents the reverse transcription step. So it prevents the integration of the viral genome into the host cell genome. So what it would do is if I would just transduce my cells, I wouldn't expect ORF8 to actually be stably integrated into the genome of these cells. But we can counteract SAMHD1 with this VPX protein. So this is a protein from simian immunodeficiency virus, which is a retrovirus that infects non-human primates. And what it does is it's able to use the host cell E3 ubiquitin ligase system to target SAMHD1 for proteasomal degradation. And as a result, it would allow for infection and it would allow for infection in these THP1 cells or in monocytes in general. So in order to transduce my THP1 cells, then I would have to make two sets of particles and then co-transduce. So one of my particles would have to encode VPX, my other one would have to encode ORF8. I then have to infect my cells with both these viruses, and only then would I expect my cells to be able to integrate the ORF8 encoding DNA, and only then would I be able to produce a stable cell line encoding ORF8. So when designing your own viral vector experiments, it's really important to also consider whether there's any restriction factors in these cells that may inhibit infection with the virus you're trying to use. So in conclusion, viral vectors are really an invaluable tool for genetic modification, and they've really established their place in biomedical research, gene therapy, and vaccines. The choice of the vector that you use has to be tailored to your application, and it depends on the size of your transgene, the type of expression you want, so transient or stable, as well as your target tissue. And while I presented a lot of attempts to try and use these vectors to treat many diseases, progress really has to still be made in this field in order to unlock their full potential as biologics. So thank you all for your attention. I hope this talk inspired you to use virus vectors in your own work. And if not, I hope you learned something either way. So thank you.
Thank you so much, Ariana. That was an incredibly comprehensive and really clear presentation. So thank you for that. We have time for questions. Does anyone have anything for Ariana? You can use the chat or you can unmute yourself.
Thank you very much, Ariana. It was really, really clear and like really extensively clear and useful. Just a quick question about your project at the end that you were talking about.
Yeah.
You said that this protein, the ORF8, is in the bloodstream as well. Mm-hmm. So, but you think that the downregulation of MHC class 1 is intracellular? My question is that basically, why don't you just wash this protein onto your cells instead of transfecting them?
Yeah, so that's an aspect that we're looking at. So we're looking at both downregulation intracellularly and downregulation secreted. So we've, for example, transfected the cells and then harvested the supernatants, and we've tried to treat THP1 cells, doing binding assays, for example, looking at the interaction between ORF8 and Fc gamma receptor 1a. It seems that the secreted doesn't affect it as much, but monocytes can actually be infected by SARS-CoV-2. So it's thought that this replication, it's like an abortive infection, but they can be infected. So there is the possibility that they can be infected by SARS-CoV-2, and then ORF8 would also be expressed intracellularly. So there's kind of both aspects or both sides that we're looking at. But that's also one of our questions as well. What's the effect of secreted ORF8?
Very cool. Thank you.
Yeah.
Hi, Ariana. Your presentation was really good. it. It was amazing. I just have a question. It's regarding my project, but yeah, I work with muscle, basically myometrium cells. They're very hard to transfect. So we have already done transfection for or using shRNA to knock down, but we wanted to use CRISPR, and I've tried using transfection method for CRISPR. But what I hear is if we use transduction for CRISPR for knockout, it might keep expressing the Cas9 and it might insert more mutations. So, I mean, you didn't talk much about, but you did put up a slide saying that they are used for CRISPR-Cas9, but you didn't talk much about like the downsides.
Yeah. To be honest, I've never used virus vectors for CRISPR-Cas9 delivery. I think though adenovirus vectors are used for that because you don't want the DNA to be integrated. So if you would use like an adenovirus or maybe an AAV vector, depending on like the size of your gene, you can deliver it and then you wouldn't expect it to be like integrated. So it would just kind of be very transient. You would express your like editing machinery and then you would like expect your cells to, I guess, it would modify the genome within your cells. I'm not sure if that's helpful, but yeah.
Okay. Thank you. Thank you so much. Yeah.
Are there any other questions for Ariana? If not, I think we'll end there. Thank you again so much. You had a lot of people telling you that it was a really great talk in the chat. Yeah, thanks everyone. If you did sign up for snacks, just go pick them up in the room. It was on the Google form that you used to sign up for that. So thank you everyone for attending.
Thanks everyone.